This podcast is going to review the urinary system laboratory, and this beginning slide just diagrams the basic association between the arterial and venous components of the vasculature in the kidney in relation to the nephron. The arcuate vessels are at right angles to the interlobular vessels, and that they occur at the junction between the renal cortex and renal medulla. In the diagram you can see the proximal tubule, the distal tubule, and the thin limb of Henle's loop in the nephron proper. Here is a section of the cortex of the kidney. This is a plastic section. You can identify renal corpuscles, proximal and distal tubules, and a capsule composed of dense connective tissue. Profiles of the proximal tubule generally stain more eosinophilic than those of the distal tubule, and they're more numerous than those of the distal tubule. You might also be able to note that nuclei of the cells of the distal tubule are much more prominently stained than those of cells that make up the proximal tubules. In this section of the kidney cortex, you can easily identify medullary rays, also called the pars radiata, that contain groups of parallel tubules. These medullary rays alternate with regions containing the convoluted tubules and renal corpuscles. This region would be called the pars convoluta. Each medullary ray is considered the center of a cortical lobule, and a medullary ray contains the straight portions of the proximal and distal tubules as well as the cortical collecting duct. Here you can identify renal corpuscles and an interlobular artery running between two corpuscles. In each renal corpuscle, you should be able to identify Bowman's space, also called the urinary space, the parietal layer of Bowman's capsule, and the glomerulus, which is the capillary bed. Note in this region of the cortex of the kidney, the renal corpuscle, and the prominent view of the interlobular artery. In the renal corpuscle, you should be able to identify the glomerulus, the Bowman's or urinary space, and the parietal layer of Bowman's capsule. You can identify proximal convoluted tubules and the macula densa region of the distal tubule. Here is a much higher power view of the renal corpuscle. You can identify the glomerulus, the urinary space, and the parietal layer of Bowman's capsule. You can also see the urinary pole, that's the site at which Bowman's space becomes continuous with the lumen of the proximal convoluted tubule. You can note that the simple squamous epithelium of Bowman's capsule is continuous with the simple cuboidal epithelium of the proximal tubule. In this region of the kidney cortex, you have again a higher power view of the renal corpuscle, but you can see profiles of proximal and distal convoluted tubules. You can also note the macula densa in the distal tubule, and then lumens of the proximal tubules are almost occluded by their large cuboidal epithelial cells. Lumens of the distal tubule tend to be more open, and they're surrounded by a larger number of cells per cross section. In this image you can identify the glomerulus, Bowman's space, also called the urinary space, the parietal layer of Bowman's capsule, and the vascular pole. The vascular pole is the site at which the afferent arteriole enters the glomerulus and the efferent arteriole emerges from the glomerulus. Neither of these arterioles can be identified absolutely in this section. You can also identify proximal convoluted tubules, and you can see a brush border on some of these cells. You can identify the distal convoluted tubule, which is visible in the extreme right central portion of the field, and you can also note the macula densa region of the distal tubule that's shown near the black arrow on the slide. You can observe conspicuous numbers of red blood cells in the lumens of the capillaries associated with the glomerulus, and mesangial cells and cells of the visceral layer of Bowman's capsule, also these would be podocytes, are closely associated with the endothelial cells of the glomerulus, but you can't distinguish among these three types of cells by light microscopy. Extraglomerular mesangial cells would be present in the small group of cells that are near the black arrow, but at this level of magnification you can't pick them out with absolute certainty. In this section of the kidney cortex, you can identify a renal corpuscle, proximal convoluted tubules, and distal tubules. You can note that the cytoplasm of the proximal tubules is considerably more eosinophilic than that of the distal tubule cells. You can also note indistinct cellular boundaries in both types of cells. In the renal corpuscle, you can identify the vascular pole and an arteriole at the small arrow. It could be either the afferent or efferent arteriole. There's no way to know from this slide. And the thick arrow shows the macula densa in a portion of the distal tubule. In this region of the kidney cortex, you can identify proximal and distal convoluted tubules. The proximal convoluted tubules can be distinguished from the distal tubules because their cells are larger, they stain more darkly, and in a typical H&E section they'd be more eosinophilic in staining, and you can even note a brush border on some of the cells in the proximal convoluted tubule, and you can note at this magnification the basal striations. And these cells would be specialized for active transport, so the brush border is increasing luminal surface area. Interdigitated amongst these basal striations would be mitochondria to power active transport. This is a relatively low power view at the junction between the cortex and medulla. You can clearly see an arcuate artery and vein in cross section on this image. These blood vessels are so named because they arch along the border between the cortex, which is on the right side of the slide, and the medulla, which is on the left side of the slide. Now you should have no trouble identifying the cortex because of the renal corpuscles that you see and because of the medullary rays. And this is a good point to remind you of the fact that even though we label these structures as medullary rays, they are cortical structures, and if you want to think about them, you can think of the medullary rays as containing the straight portions of the proximal tubule, the straight portions of the distal tubule, and the cortical collecting ducts, and these cortical structures are pointing towards the medulla, hence the name medullary ray. This is a plastic section of the outer medulla of the kidney, and in this section you can identify thin limbs of Henle's loop. Those are shown at the thin arrows, and thick segments of Henle's loop, as well as collecting ducts, which are shown at the larger arrows. And profiles of the vasa recta can be shown at the arrowhead. Is a section of the inner medulla of the kidney, and in this section you can identify thin segments of Henle's loop shown at the thin arrows. You can identify at the thicker arrows sections of the largest collecting ducts or papillary ducts, this being deep in the medulla. This is where these ducts begin to coalesce. And then finally at the arrowheads you can see sections of the vasa recta, and this is nicely indicating these vasa recta sections because you can see red blood cells in the lumen of these vessels. Another section in the inner part of the medulla, you can see the papillary ducts, and you can see thin limbs of Henle's loop and sections of the vasa recta at the black arrowheads. This is a section of the ureter. You can identify the mucosa, which consists of transitional epithelium and the underlying lamina propria, and you can see a little bit of the muscularis, and just to the edge of the slide, possibly some of the adventitia. And here's a view of the male urethra. You can identify mucus glands of Urethra that are associated with this epithelium.